Hello to all. This is Priyanka Sonar, a research student at DY Patil, and today we will be discussing about new visions in infection defense. What is exactly infection and how does a beautiful structured human body deal with it? An infection is the colonization of a host by microbial species. Infecting microbes seek to use the host resource to reproduce, often resulting into diseases which can range from mild to fatal. And which are the infection causing agents? They are viruses, prions, bacteria, viroids, and even larger organisms like fungus and, in some cases, macroparasites. The following detailed picture gives us a glimpse of the variety of microorganisms that can affect human bodily organs. Hosts, in this case, our human body can fight these infections by using the immune system. But new vision of dealing with infection defense is basically the idea of bringing in new technologies to prevent cross transmission of pathogens, often at significant financial cost and unique implementation consideration. There may be an adjunctive role of such technologies in existing infection prevention programs as a part of multifaceted approach. Talking of technologies, I think COVID 19 actually taught us how to use a whole new set of techniques to prevent infection. Thus, we incorporated new technologies to improve cleaning monitoring, started making use of fluorescent markers in them. Now we have touchless technologies, even room cleaning robots. Talking about surfaces, we often touch door handles, able surfaces, etc. Several antimicrobial coatings are under preclinical studies for their potential applications to surface in healthcare centers. Of these coatings, copper has been studied most extensively. Hand hygiene monitoring new technologies have also emerged in recent times. It is one of the core infection prevention strategy that is a simple technique to perform which can help us to prevent many infectious disorders. Studies have estimated a wide range of compliance with an average of roughly 40% according to World Health Organization. And I think it's a pretty good number to start moving towards a world where we can treat better and make friends with these little creatures called microbes, which can be good and can be bad. This is my view on new visions of dealing with infections, and thank you for listening.
Hello everyone. Myself, Sai Bharat and Raja Pawan Karthik, presently pursuing undergraduate in artificial intelligence in Amrita Vishwavidyapeetham, Kerala. Our topic of research is oral cancer detection using machine learning. Basic technologies. In our project, oral cancer refers to the cancer development around the part of the mouth, including lips, gums, tongue, and uh, floor of the mouth, and also through the throat of the mouth, where oral cancer is very common in nowadays. People who use tobacco, consume alcohol aggressively, have a low diet of fruits and vegetables, and those who have family history of cancer may lead to this oral cancer. Machine learning. Here we are trying to use machine learning programs with the help of artificial intelligence, which enables us to compute and learn about the data and improve the performance over time without being programmed. In other words, machine learning algorithms that we are using is automatically identifying the patterns, areas from the data they are given in order to make predictions and decisions where the cancer is found or not. Dataset. We are presently trying to get the dataset from the Kaggle datasets where the clinical dataset is present. Some of them are cancerous and some of them are non-cancerous. We have some limitations from the Kaggle dataset that we have gained. So we have tried to do data augmentation for now. As we are working on the data augmentation, we have gone through various uh, problems such as insufficient data, like over efficient cancerous dataset and non-cancerous dataset is very less, and we need around 1,000 dataset for the predictions, but we have around below 100. So we have to do pre-processing the data to get the present accurate machine learning program. So we have tried to exhibit various types of data set from the given data set, like decolorizing, image enhancement, flip, rotate, and mirrorizing horizontally and vertically. So we have gone through several data sets and we have taken some of them with the help of some image restoration and other techniques using Python. We especially thank our mentors, Dr. Prashant, Dr. Pramod, Professor Jayaraman, and Sundar Rajan. I would like to thank Tanvi, Professor Kavikishore, Dr. Sugunakar, Adi, Sneha, Girik, who has been part of this project. And a special thanks to Mata Amrutanandamayi, the Chancellor of Amrita University. Thank you.
My name is Aditya Satish. I'll be presenting on the topic data imbalance and multi-level classification. For a machine learning algorithm to perform very well, we need data. Data is a crucial part for machine learning. So we have balanced data and imbalanced data. Consider if you are having two classes, class A and class B. If the number of samples in class A is approximately equal to number of classes in class B, then the data is balanced. If there is a minority class or majority class in the data, then data imbalance occurs. This is an example for balanced data and imbalanced data. In this case, if you can see, this is a real world scenario. In this case, the total number of samples in class A and class B is approximately equal. But if in this case, this is a test case for cancerous genes. Out of maybe if you are checking for people with cancerous genes, only 10% will be cancerous. This is a data imbalance. This is a real world issue. This is my data. This FASTA sequence is my input, and other 9 features are my output. So this is a problem faced in my data set. If you can see that the shortest length of my sequence is 16 and the largest length is 5559. So my length of the sequence varies from 16 to 5559. Coming to the main part, the imbalance. This is my data and this is the imbalance on each of the classes in my data set. In each of the 9 features I'll be handling imbalance. In my data, FASTA sequence serves as input and the 9 features are my output. I have converted the FASTA sequence into ASCII numerical and make it as numbers, and I have 0 point all the sequence at the maximum length of 10,000. These are the techniques for handling data imbalance issue. Here I will be talking about random undersampling. Undersampling basically brings the minority class and majority class equal by decreasing the minority class data. And here we have oversampling. Oversampling basically means that duplicating the minority class and make the copies of minority class and make the majority and minority equal. This is another technique, synthetic minority oversampling technique, SMOT. Here in the first image, we can see we have minority classes, and this algorithm looks for choose any of the data set and choose the nearest neighborhood of that k nearest neighborhood. In this case, it has chosen 5 nearest neighborhood, and it creates a straight line, and it randomly creates synthetic minority sample on that line, on the straight line. So here we are not duplicating that data. We are creating the samples synthetically. This is the algorithm for SMOT.
My name is Sneha. I'll be presenting on the topic biologically inspired chaos and architecture for hypothetical protein classification. Hypothetical proteins are those proteins whose structure or function informations are unknown but are assumed to be.
Razul, is this video abstract audible to audible, right? Okay, okay. Yeah, thank you.
be expressed nowadays. And many hypothetical proteins are assumed to be involved in crucial biological processes, and a lot of researchers are going on to figure out what they are and their roles in these processes. So, in my paper, I classified hypothetical proteins using an artificial neural network called CHAOSNet, which was created in 2019 to make use of some of the best features of biological neural networks, that is, a property existing in the biological neural networks called CHAOS. As an attempt to use one of the best properties of biological neural networks arising as a result of which chaotic behavior of neurons, CHAOSNet was developed with one-dimensional chaotic maps generalized Lorentz series as its individual neurons. This is the proposed architecture of CHAOSNet where the input neuron will, in the input layer will have n GLS neurons, and after extraction of topological transitivity symbolic sequence feature, then it will move on to the output layer, and later for testing the cosine similarity of the values inside the output layer and the topological transitivity symbolic sequence features will be calculated for prediction. The dataset was taken from the paper model functional hypothetical proteins through an endpoint classification scoring schema, and there are 100 data instances and 9 features, and the features are listed here. And in the CHAOSNet, there were a few hyperparameters, which are the neural activity B, which was taken as 0.89, the discrimination threshold Q 0.49, and the epsilon value defined for the neuron, which is 0.043. So, coming to the results and discussion, we have compared the proposed CHAOSNet algorithm with the results obtained in the paper using machine learning algorithms like decision tree, net bias, MLP, support iteration, etc. And also our confirmed cross-validation is done on the dataset before each algorithm is executed. And we can see that for 6-point scoring schema, CHAOSNet performed very well as compared to other classifiers, and in 9-point classification, it was comparable. And one of the main properties of CHAOSNet is that it can be trained with very few samples per class. So when it got trained with only one sample per class, the accuracy was above 80%, and when it got to 20 samples per class, it got to about 96%. And feature selection was performed to obtain best features for CHAOSNet, and we can say that CHAOSNet performed to have accuracy 96.25, and the others performed much less. Hence, we can say that CHAOSNet outscored other algorithms for 6 features, while the results were close for 9 features. And if we employ multi-layer CHAOSNet with fully connected layers, then we might be able to improve the performance. That's all. Thank you.
Hello everyone, this is Ranjana from Amrita School of Biotechnology. I'm pursuing my master's in bioinformatics and currently in my final semester. I'll be talking about my dissertation topic, that is benchmarking an exome pipeline for inferring consensus variants. My project guide is Dr. Prashant Surajala, a principal scientist, Amrita School of Biotechnology. So first of all, benchmarking is a process of evaluating the performance of a pipeline by comparing the results to a known or expected set of results, usually derived from gold standard dataset or reference databases. Variant calling is a crucial step in the analysis of genomic data, which involves identifying differences between the DNA sequences of different samples or individuals. However, different variant calling tools may produce different results due to variations in algorithms, parameters, and thresholds used. So we will be using various variant calling tools such as VT, WASCAN, FreeBIAS, and VCF tools to generate VCF files with raw variants, which will be going through downstream processing, and we will be focusing on this intersection of variants. So finding variants through the intersection of variants involves taking the variants that are present in all the variant calling tools used in the analysis. So in the pipeline workflow, we'll be working with bow tie that is used to align the forward and the reverse tree to the reference sequence that generated SAM file. Then we'll be using SAM tools, which converts SAM file to BAM format, sorts it and indexes it. Then we have the Picard tools, that it's a toolkit which contains a mark duplicates tool. The PCR duplicates can occur during library preparation when multiple copies of the same DNA fragment are amplified by PCR and sequenced. These duplicates can lead to biases in the downstream analysis, such as variant calling and differential expression analysis. So, the mark duplicates tool identifies and marks these duplicates in the BAM file based on the alignment coordinates, sequence, and orientation. At last, we have variant callers. Various variant callers we have been using, such as the VT, WASC, and FreeBias and VCF tools. These are used further as variant callers to make a final VCF file. We have the GATK4 workflow as our validation workflow. In this also, we have GATK is a genome analysis toolkit, which is widely used software package for analyzing high throughput sequencing data, particularly for variant calling and quality control. So the base quality is in the line reads are recalibrated to account for systematic errors in the sequencing and element processes. This step uses a machine learning model that learns from known variants in the data and from the reference genome. At last, we have the GATK haplotype caller, sensitive method for calling variants. This tool generates a likelihood model for each possible variant allele, taking into account the local haplotype structure, sequencing error rates, and other factors. In the discussion and future aspect, we can say that variant calling tools usually differ in algorithms, filtering strategies, and also in the output. High sensitivity is always accompanied by low precision and vice versa. So the influence of variant coverage and background noise on variant calling are a common issue that these NGS-based analysis suffers. So we are focusing on finding those consensus variants that show high probability of occurrence in the sample. And that can be done by the use of gold standard datasets from 1000 Genome Project or Genome in a Bottle Consortium for further validating and reporting those rare variants. Thank you.
Good morning, Honorable. I'm Anjali Krishna, currently doing PhD first year under the supervision of Dr. Prashantan Surajala and co-supervisor Dr. Renuka Surajala. So my topic is inferring variants of unknown significance in rare disease genetic. It's an all Indian-centric studies. So first of all, I'm taking the rare disorders. So first question is, are rare diseases are rare or not? So based upon the literature review, you can see in US, black peoples are already affected some kind of rare disorders, and 1 per 10,000 in Taiwan. If we take an India had 85 million population or 76 to 85 million population. So almost approximately 7.8% populations are tagged as a rare. So mostly rare disorders are usually monogenic and largely heterozygous, and the important factor is manual frequency. It's less than 0.01 for rare disorder, and some extremely case is less than 0.001. And it have many uncharacterized mutations, and these mutations are largely variants of unknown significance. So the parallel genotypes are not necessarily transcended to the children, which means there are two types of mutation. It could be rare or de novo mutations. And rare disease variants in congenital pouch colon and anorectal malformations anomaly were discovered. That means like we have two samples like congenital pouch colon and anorectal malformation. So the congenital pouch colons is a condition of the shortened and pouch-dilated colons, and they usually associated with the anorectal malformation. And the pathogenesis of the embryology of congenital pouch colons are not well understood, but dietary environmental factors that may be affected or contributing factors in this pathology. Then it's mostly visible in India than the other countries, with reviewers showed like 90.2%. And here some of the papers are give us the references. So here you can refer like 4 papers which elaborate the case study. And our lab has also worked the inferring rare variants in aplastic anemia, aquatic aplastic anemia. The main symptoms are fatigue, fever, headache, dizziness, and shortness of breath, unexplained of easy bruising, rapid or irregular heart rate, etc. So the important thing is the missing link between the rare diseases. So rare diseases are like maybe it leads to autoimmune disorder or infectious diseases, neurological disorders, or risk factors risk factor or prevention. So if we find the missing link between the diseases, we can easily provide the treatment, or we can easily find the kind of treatment or treatment kit or what. And the problem statement in this problem, like the thing is like growing myths and challenges that may be that many rare disorders are autosomal dominant or recessive. So no knowledge on variants of unknown significance and lack of the consensus and genetic test hint a detection of rare disease. So does rare disease have preferences for any specific allele or not? So these are the research gaps or the problem statement which we have to solve. So my main objectives, based upon these, my main objectives are kind of like to check uh, whether or not genetic variation is best seen in non-coding RNAs, or to identify the role of variants of unknown significance in rare diseases and establish causal relationship between variants in the genetic of rare disease common to India, to identify the genetic susceptibility of variant of unknown significance, finding unique roles in their pathogenesis. And the last one is to dock the variant of unknown significance emerging from the rare phenotypes with targeted ligands and establish causal relationship with disease causal genes. And here I wrote the PRISMA guidelines. Here you can see. And coming to the expected outcomes, so establishing causal relationship for non-coding RNAs and identifying genetic variations in them in some selected rare disease of India, and developing a standard operating procedure or framework for rare disease and India context, taking variant of unknown significance and attempting to understand long range sequencing chemistry using OND-Minion for identifying rare exomic variants and finding the roles of interfacial or causal interfacial, that means the causal variants residues across the rare disease. That's all. Thank you.
Hello everyone, my name is Bhargavi. I'm a PhD scholar at Amrita School of Biotechnology. My presentation for today is titled Integrated System Genomics Approaches for Deciphering Candidate Genes that are Regulating Prostate Cancer Phenotype in India. A prostate cancer is a adenocarcinoma that develops in the glandular part of the prostate gland. The major risk factors for developing prostate cancer are age, ethnicity, obesity, family history, lack of exercise, persistent elevated testosterone levels, etc. I have chosen prostate cancer for my research because there is a serious gap in the knowledge base of candidate genes that are involved in prostate cancer phenotype, particularly in the Indian population. There also seems to be a missing link between diabetes and prostate cancer. Prostate cancer is a fairly common cancer, being the second most prevalent cancer worldwide and the third most prevalent cancer in India. Um, as part of the CAPSI consortium of India, which is the only consortium that deals itself with whole exome sequencing of prostate cancer samples um, and performing genomic analysis. A pilot study was conducted by Gupta and Shukla et al. In 2020, where they uncovered mutations in genes that are associated with obesity, diet, mitochondrial dysfunction, and vitamin D deficiency in a prostate cancer cohort from India. The mutations identified are represented in the picture on the right. The objectives of my research include the identification of lead mutations that are influencing diabetes in a prostate cancer cohort from India and performing whole exome sequencing to identify the variants and also find possible links to diet and lifestyle. Um, I also aim to identify candidate differently expressed genes through including long non-coding RNAs and checking their regulatory nature using a fly model. Pictured here is the benchmark pipeline for our whole exome sequencing, and the variants identified through this will then be validated using a fly model where we're using axillary gland and Drosophila as a model to study prostate cancer. Axillary gland and Drosophila is a functional equivalent of the prostate gland, hence this will be used for the transcriptomic analysis. A cell type specific transcriptomic analysis can be achieved without RNA isolation using the targeted DAM-ID method, where we will be isolating methylated DNA for fault to occupancy, and the gene expression profile obtained uh, uh, will be able to provide us information of differentially expressed genes. So the comparison of the transcriptome of wild-type axillary gland and the proliferated axillary gland uh, will shed some light on the overexpressed or underexpressed genes in both cases, and this will then be screened through our candidate genes that have been identified through whole exome sequencing in the prostate cancer cohort in humans. Through this work, we are hoping to identify risk SNPs that are associated with prostate cancer in India, identify a possible association of diabetes with prostate cancer, and also identify cancer genes that are expressing both in the prostate gland as well as in the axillary gland, and also perform functional analysis of those genes to identify protein products or RNA products. These are some of the references. Thank you.
Namashivaya. We are final year students from Amrita School of Biotechnology, Amritapuri, Kerala. We are currently doing a project under the guidance of Dr. Prashant Survajla, and the project is ascertaining the common denominators for rare disease genomics. I am Nayantara, and there are 6 others with me who are doing this project: Arya, Midhun, Dipankar, Meenakshi, Megha, and Vrinda. Now, what exactly is a rare disease? As of now, there's no universal definition. It is generally a health condition with a low prevalence when compared to other common diseases. There is no proper cure or treatment protocol for a majority of rare diseases.
To analyze rare disease are Phenylaser, GeneCards, CleanWall, GeneMania, String, along with other databases such as NCBI. My name is Deepankar Dilip, and my rare disease of interest is Fabry disease. Fabry disease is an X-linked lysosomal storage disorder caused by a mutation in GLA gene. GLA has 7 exons and is located at XQ22 region. It's sometimes also known as Anderson-Fabry disease.
And the disease that I've chosen is X-linked adrenal leukodystrophy. It is an X-linked peroxisomal storage disease which leads to the accumulation of fatty acid, causing the toxicity of adrenal cortex.
And Santosh, and I have taken Addison's disease. Addison's disease is a primary adrenal insufficiency resulting from the bilateral destruction or dysfunction of the adrenal
cortex. Is Gaucher's disease. Gaucher's disease results in the buildup of fatty substances in organs like spleen and liver. It causes these organs to enlarge and affect their function. These fatty substances can also build up in bone tissues, weakening the bone and increasing the risk of
the rare disease, von Willebrand disease, which is autosomal inheritable bleeding disorder in which the blood doesn't clot properly, and it is caused due to the deficiency in von Willebrand factor.
And the rare disease which I've chosen is Duchenne muscular dystrophy. It is generally a progressive muscular disease, and it is because of a mutation in a gene called dystrophin, and it usually starts in young boys at the age of...
And my rare disease of interest is pulmonary fibrosis. It is a lung disease that occurs when lung tissue becomes damaged and scarred. So the symptoms of these are shortness of breath, a dry cough, fatigue, etc. Medications for the same are chemotherapy, drugs, and heart medications.
Systems genomics integrated approaches are very much needed to find pathogenic variants in diseases associated with all the 7 diseases which we've mentioned above. Namashivaya. We are final year students from Amrita School of Biotechnology, Amritapuri, Kerala. We are currently doing a project under the guidance of Dr. Prashant Survajla, and the project is ascertaining the common denominators for rare disease genomics. I am Nayantara, and there are 6 others with me who are doing this.
I am Sana Fatima from Telangana Mahila Vishwavidyalayam. I am here to shed light on application of artificial intelligence in agriculture. According to United Nations prediction data on population and hunger, the world population will increase by 2 billion people by 2050, requiring a 60% increase in food productivity worldwide. While traditional farming methods are failing to meet this huge demand, the industry is making use of artificial intelligence technology to overcome agricultural problem. Artificial intelligence deals with smart machines capable of performing tasks employing human intelligence. Machine learning is the main theme of artificial intelligence. It uses algorithms and statistical models to convert raw data into classified data to make predictive decisions. Machine learning along with artificial intelligence can improve field mapping and yield prediction of crop. Presently, smart irrigation technique have been developed using Internet of Things. It is a method that uses information from Internet and uses it in a global network of software and sensors. Sensors connected to microcontrollers are installed in the field. These sensors detect any changes in the environmental factors such as temperature, soil moisture content, humidity, soil pH, and carry out the needful action. Drones work in combination of both sensors and GPS. They are furnished with different sensors for digital imaging, spraying pesticide, field analysis, and crop monitoring. Drones are being widely used in almost every aspect of agriculture, such as crop health assessment, remote sensing, irrigation, crop monitoring, spraying pesticides and fertilizers, soils and field analysis. Artificial intelligence employs image processing technique to detect nutrient deficiency, color of plant, shape of leaf, seed, texture, etc. It involves steps like image input, pre-processing, feature extraction, classification of extracted feature data, and gives an output. Another application of artificial intelligence in agriculture is in terms of agrobots. These agrobots are working on field and becoming an integral part of present-day precision agriculture and digital farming. They are performing various operations like preparation of land, crop scouting, sowing seeds, weeding, pest control, transplanting seedlings, and harvesting crop without any human involvement. In this era of information and communication technology, the smartphone apps are being used in agriculture. These apps provide a climatic and seasonal data. They also provide a platform for farmers to seek solutions for problems faced in the field. ICRISAT has developed a sowing app in collaboration with Microsoft. These apps have revolutionized modern agriculture to a greater extent. Based on their uses, these are classified as agri-crop mobile application, multi-informative application, calculative mobile application, diagnostic mobile application, agri-academic application. Blockchain technology stores and manages data, record information about the state of farm, supplies and contracts in agricultural sector. It maintains all the related in account and transaction securely. Blockchain technology along with Internet of Things increases dependability and